The basic rhythm of breathing is controlled by respiratory centers located in the brainstem. This rhythm is modified in response to input from sensory receptors and from other regions of the brain. Your goals for learning are to understand how the respiratory centers control breathing to maintain homeostasis, to examine how PCO2, pH, PO2, and other factors affect ventilation, to understand the relationship between breathing and blood pH, to explore the factors which stimulate increased ventilation during exercise. Here's what you need to know: the basic principles of homeostasis, the general anatomy of the brain, the role of the inspiratory and expiratory muscles, the definition of pH, how to interpret the oxygen-hemoglobin dissociation curve. To review the breathing muscles or the oxygen-hemoglobin dissociation curve, click the appropriate link button. To see definitions of terms, click the bold red words. The control of respiration is tied to the principle of homeostasis. Recall that the body maintains homeostasis through homeostatic control mechanisms, which have three basic components: receptors, control centers, and effectors. Let's see how this applies to the respiratory system. The principal factors which control respiration are chemical factors in the blood. Changes in arterial PCO2, PO2, and pH are monitored by sensory receptors called chemoreceptors. The chemoreceptors send sensory input to respiratory centers in the brainstem, which determine the appropriate response to the changing variables. These centers then send nerve impulses to the effectors, the respiratory muscles, to control the force and frequency of contraction. This changes the ventilation, the rate and depth of breathing. Ventilation changes restore the arterial blood gases and pH to their normal range. The basic rhythm of breathing is controlled by respiratory centers located in the medulla and pons of the brainstem. Within the medulla, a group of neurons in the ventral respiratory group sets the basic rhythm by automatically initiating inspiration. Click the inspiratory neurons to see how they function. The inspiratory neurons send nerve impulses along the phrenic nerves to the diaphragm and along the intercostal nerves to the external intercostal muscles. Click the inspiratory neurons to see how they initiate inspiration. The nerve impulses to the diaphragm and the external intercostal muscles continue for a period of about 2 seconds. This stimulates the inspiratory muscles to contract, initiating inspiration. A second group of neurons in the ventral respiratory group now fires, inhibiting the inspiratory neurons for about 3 seconds, which allows the muscles to relax. The elastic recoil of the lungs and chest wall leads to expiration. The automatic rhythm generated by these two groups of neurons, alternately inhibiting each other, produces a normal resting breathing rate ranging between 12 and 15 breaths per minute. Click the inspiratory neurons to observe normal breathing. The dorsal respiratory group, or DRG, acts as an integrating center for peripheral stretch and chemoreceptor inputs and influences the activity of the neurons in the VRG. Both the DRG and the VRG receive inputs from other respiratory centers in the pons, which modify inspiration and allow for smooth transitions between inspiration and expiration. Although the basic rhythm of breathing is established by the respiratory centers, it is modified by input from the central and peripheral chemoreceptors. They respond to changes in the PCO2, pH, and PO2 of arterial blood, which are the most important factors that alter ventilation. Click the central chemoreceptors button to see their location. The central chemoreceptors in the medulla monitor the pH associated with carbon dioxide levels in the brain. The chemoreceptors synapse directly with the respiratory centers. Now click the Peripheral Chemoreceptors button to see their location. The peripheral chemoreceptors are found in two locations: the aortic bodies within the aortic arch, and the carotid bodies at the bifurcation of the common carotid arteries. The peripheral chemoreceptors monitor the PCO2, pH, and PO2 of arterial blood. This information travels to the respiratory centers via the vagus and glossopharyngeal nerves. Click either button to review the location of the chemoreceptors. Let's look more closely at how the central chemoreceptors respond to carbon dioxide, the most important factor controlling the rate and depth of breathing. Click the central chemoreceptors to see their response. Now, let's look at the role of the peripheral chemoreceptors. Increased carbon dioxide levels in the arterial blood result in decreased blood pH, which stimulates the peripheral chemoreceptors. They respond by sending more nerve impulses to the respiratory centers, which stimulate the respiratory muscles, causing faster and deeper breathing. More carbon dioxide is exhaled, which drives the chemical reaction to the left and returns the PCO2 and pH to normal levels. Click the blood vessel to learn how the peripheral chemoreceptors respond to other arterial pH changes not caused by carbon dioxide. The peripheral chemoreceptors also respond to acids such as lactic acid, which is produced during strenuous exercise. Active muscles produce lactic acid, which enters the blood, releases hydrogen ions, and lowers the pH. As we have just seen, the decreased pH stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers, which stimulate the respiratory muscles to increase the breathing rate and depth. More carbon dioxide is exhaled, lowering the PCO2 in blood, driving the chemical reaction to the left, and lowering hydrogen ion levels. Click the blood vessel to see the flowchart again. The peripheral chemoreceptors also monitor arterial PO2. However, the arterial PO2 must drop below 60 millimeters of mercury before the chemoreceptors respond. Click the graph to learn how the oxygen-hemoglobin dissociation curve explains this phenomenon. What changes will occur if a person hyperventilates, that is, breathes deeper and faster than necessary for normal gas exchange? Click the correct combination of buttons to indicate the changes in the blood that will result from hyperventilation. Refer to the chemical equation for help. No, try again. Low PCO2 is correct. However, low pH is incorrect. Correct. During hyperventilation, carbon dioxide is exhaled, lowering the PCO2. This drives the chemical reaction to the left, decreasing the hydrogen ion concentration and increasing pH. Since the PCO2 is low, the central chemoreceptors send fewer impulses to the respiratory centers. Since the pH is high, the peripheral chemoreceptors also send fewer impulses to the respiratory centers, which send fewer nerve impulses to the respiratory muscles, thereby further decreasing breathing rate and depth and returning the arterial gases and pH to normal levels. Click the blood vessel to see the flowchart again. This chart summarizes how the three major chemical factors, PO2, pH, and PCO2, modify breathing rate and depth. Click a button to review each pathway. When the PO2 drops below 60 millimeters of mercury, the peripheral chemoreceptors send nerve impulses to the respiratory centers. The respiratory centers send nerve impulses to the respiratory muscles, increasing ventilation. More oxygen is inhaled, returning the PO2 to normal levels. When cells release acids into the blood, the acids release hydrogen ions, which lower the pH. This stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers. They, in turn, send more nerve impulses to the respiratory muscles, increasing ventilation. More carbon dioxide is exhaled, which returns the pH to normal levels. An increase in PCO2 leads to a decreased pH in the blood, which stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers. In addition, the increased PCO2 leads to a decreased pH within the brain. This stimulates the central chemoreceptors to send more nerve impulses to the respiratory centers. The respiratory centers send more nerve impulses to the respiratory muscles, which increase breathing rate and depth. More carbon dioxide is exhaled, returning the PCO2 and pH to normal levels. In addition, several other factors influence ventilation. These factors include voluntary control, pain and emotions, pulmonary irritants, and lung hyperinflation. Click the buttons to learn more about each factor. Pain and strong emotions, such as fear and anxiety, act by way of the hypothalamus to stimulate or inhibit the respiratory centers. Laughing and crying also significantly alter ventilation. By sending signals from the cerebral cortex to the respiratory muscles, we can voluntarily change our breathing rate and depth when holding our breath, speaking, or singing. However, chemoreceptor input into the respiratory centers will eventually override conscious control and force you to breathe. Dust, smoke, noxious fumes, excess mucus, and other irritants stimulate receptors in the airways. This initiates protective reflexes such as coughing and sneezing, which forcibly remove the irritants from the airway. Stretch receptors in the visceral pleura and large airways send inhibitory signals to the inspiratory neurons during very deep inspirations, protecting against excessive stretching of the lungs. This is known as the inflation or Herring-Breuer reflex. Now let's look at the changes in ventilation during exercise. Ventilation increases during strenuous exercise, with the depth increasing more than the rate. Remarkably, it appears that changes in PCO2 and PO2 do not play a significant role in stimulating this increased ventilation. Although the precise factors which stimulate increased ventilation during exercise are not fully understood, they probably include learned responses, neural input from the motor cortex, receptors in muscles and joints, increased body temperature, circulating epinephrine and norepinephrine, and pH changes due to lactic acid. Click a button to explore these factors. Ventilation increases within seconds of the beginning of exercise, probably in anticipation of exercise, a learned response. It also increases because the motor areas of the cerebral cortex, which stimulate the muscles, also stimulate the respiratory centers. Proprioceptors in moving muscles and joints stimulate the respiratory centers, as does an increase in body temperature. Circulating epinephrine and norepinephrine secreted by the adrenal medulla stimulates the respiratory centers. Lactic acid, produced by exercising muscles, is another stimulus. Here's a summary of what we've covered. The basic rhythm of breathing is set by the ventral respiratory group located in the medulla. Other respiratory centers located in the medulla and pons also control breathing. Chemoreceptors monitor the PCO2, pH, and PO2 of arterial blood and alter the basic rhythm of breathing. Carbon dioxide, reflected by changes in pH, is the most important stimulus controlling ventilation. pH changes due to metabolic acids also alter ventilation. Oxygen stimulates ventilation only when the blood PO2 is very low. Other factors, such as voluntary control, pain and emotions, pulmonary irritants, and lung hyperinflation, also play roles in controlling ventilation. The control of ventilation during exercise, while complex and not fully understood, involves multiple inputs including chemical and neural factors. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.